Okay, good morning, everyone. This is our April version of EMX. And as always, we have two great and exciting speakers whom we've separately known for several years. So we will start with the biology talk as usual. And Professor Hua Chu will introduce Stephen to give his presentation, and then we'll continue with the physical sciences talk by Professor Wolfgang Jaeger from Germany. So Hua, let me pass it over to you, please.
Okay, good morning, Bob and everyone, and good afternoon and evening wherever you are. It's my great honor today to have Professor Stephen Duguid to be our speakers. Steve started his research in X-ray diffractions of membranes when he was a graduate student after he got his physics training at Caltech. And he saw the light that perhaps electron is a good thing to do structures. So in the early 2000s, he single-handedly developed a very user-friendly software for single particle reconstructions. His software has benefited the early stage of the development of single particle CryoEM, that eventually he was among the first to show that single particle CryoEM can solve atomic or near atomic resolution structures without crystals. And Steve has been very instrumental in the software development, and he has been very generous with software. He always believed in the open source code, so there are lots of users around the world actually use this software for many projects that publish in many nice papers. And he's currently the Charles Bell Professor of Structural Biology at Baylor College of Medicine, and it's my delight to have Steve to come to talk to us. His latest development, both in single particle cryo-EM as well as electron tomography using the artificial intelligence approach. Steve, welcome.
Thanks very much, Hua. Thanks for inviting me to come and give this talk. It's nice to have these opportunities to give talks to really large audiences virtually, even if we don't get to see people in person. Let me go ahead and share here. Okay. All right. So I debated a little bit what to talk about today, and I thought with the all of the recent discussions around AlphaFold and that sort of thing, that I might give a little bit of a context talk today. So, you know, if you look at sort of the traditional structural biology field, right, we have a variety of different techniques that we could apply. And then, of course, recently sort of AlphaFold has sort of stuck its way in here in coming up with putative models for pretty much any protein that you might want to come up with. Now, you know, of course, AlphaFold isn't perfect. It's not going to give you models for everything. But it at least does a pretty good job, and you know, has had a lot of structural biologists kind of worried, oh, yo, what is there still left to do? So I want to spend a little time today sort of talking about that whole context of structural biology and how we really should be moving sort of more in the cellular direction anyway. And that while structures like AlphaFold, you know, what AlphaFold can provide, and even crystal structures, high resolution cryo-EM structures, are an important starting point, foundation for our analysis. It's really not answering the final questions. It's really just giving us a starting point. So I'm going to start with a couple of just biological examples, and I'm not going to go through these in any great depth. I'm just going to use them as a sort of a tool in this discussion. The first thing I'm going to talk about is this ACR-AB Toll-C complex, which was started with a collaboration between Hua Chu and Ben Luisi doing sort of high-resolution single particle work about a decade ago. And then after Hua moved to Stanford, his postdoc, Xiao Wang, became a faculty at Baylor and sort of continued the project, but moving it more of a cellular direction. So these ACR-AB Toll-C complexes live in between the inner and outer membranes in bacteria, and are one of the complexes that's responsible for antibiotic resistance in bacteria. Basically, these are pumps, and they eliminate all of the antibiotics from the periplasm before they have time to get into the cell and kill the bacteria. So understanding these pumps has some real biological import. And the overall sort of structure that consisted of 3 different molecules, which were forming this tripartite complex, has been known for a long time. We even have crystal structures for the individual domains. Of course, there are some truncations and that sort of thing, but we have pretty decent crystal structure. And we say, well, we've got crystal structures, we kind of know how it works. Surely we must know everything about how this channel works. Well, not quite. Don't entirely know. I mean, if all you have is the crystal structure and this sort of general idea, you don't really know what the stoichiometry is between these different bits. We don't really understand how it works still. How do these 3 pieces put together actually function to pump antibiotics out of the cell? So that led to the single particle structure. The single particle structure, which they published in 2017, identified the stoichiometry of the various components, and it sort of showed what the overall assembly looked like. How was this related to this? How were the various pieces docked together? But still, that's not the complete picture. So then we move into the more cellular context. So what we're looking at here is E. coli. This is a tomographic reconstruction of E. coli. So the entire E. coli cell was frozen, tilt series was collected, and then we did this tomographic reconstruction. So we just sort of rock this back and forth a little bit. So the Toll-C complexes live in the periplasmic space here, in between the inner and outer membranes. So we can see these particles. If we look carefully, we can see the individual particles. They're sort of packed in here between the 2 membranes. Now, one of the nice things about drug efflux pumps is if you expose a cell to bacteria, they sort of automatically overexpress in the cell. So it's not very difficult to get these things to appear. Of course, you can also trigger overexpression via the common mechanisms as well. But overexpression is sort of the natural state for these things to exist in when they're active. So you can see here all of these individual Toll-C complexes sticking out of the cell. This is what they look like from the top view. They're sort of narrow and long, so they just look like little circles in there. And then we can sort of reconstitute them back into the particle. Once we've solved some sort of a low resolution structure, we can put them back in, and you can just sort of see where they all are decorating around the cell. So you can see these things are just everywhere around the outer membrane of the cell. So what can we get by doing this sort of subtomogram averaging? So instead of purifying the molecules and doing single particle analysis in vitro, we'll take the whole cell and we'll pull the particles out of the cell and then average those together and do subtomogram averaging, and we get some additional information. From this initial experiment, this was sort of the first attempt, low resolution. This was published back in 2019. We can see that a little bit about the assembly pathway of the complex, right? So you can see here between the inner membrane and the peptidoglycan layer, ACR-A and ACR-B are found without Toll-C a significant fraction of the time. Now, I'll note that this is just a chunk of membrane that got extracted along with the Toll-C. This isn't actually part of the Toll-C complex, that's just membrane. So basically, you see these partially assembled complexes and the fully assembled complex. So that already gave us at least a little bit of insight into what might be going on inside the cell. But of course, we continued. We developed the subtomogram averaging pipeline in EMAN2 much more over the years just before the pandemic. And that allowed us to achieve a much higher resolution structure. So now we have the single particle reconstruction. We had this low resolution subtomogram average, and now we have a high resolution subtomogram average. It's not quite as high resolution as the single particle structure, but that's okay, because that's not necessarily the information we're trying to get out of here. We have a crystal structure that we can dock in here too, right? So we already have good high resolution structures. The whole point here is to put it into context and understand how it's interacting with the cell. So one of the conclusions that you can see here is that this sort of pseudo six-fold symmetry that exists around the base here, every other subunit or every other pseudo-equivalent unit in the cell is interacting with the membrane. So here, in this subunit, it's actually self-interacting. ACRA is interacting with the ACRB here, but then in the next one in the sequence, as you work around the base, you can see it's actually seen to be interacting directly with the membrane. There are a variety of other things that we saw as well, including interactions with the outer membrane and that sort of thing. But the point is that by looking at it in the cell, you can actually begin to understand how it works functionally, not just what the shape is, right? Okay, so that's the first example. Now, I'd like to just sort of point this out, that the cells are really large, really complicated beasts. So this is just an optical microscopy image of a typical epithelial cell, to scale here. So about 10 microns here. So this is about a 50 micron cell. It's a pretty typical sort of mammalian cell. Now, if we wanted to solve the structure of this entire cell to sort of near atomic resolution, so if we wanted to achieve 2 angstrom resolution throughout this cell, assuming we could do it technologically, a single field of view in a current high-end electron microscope would be the size of this tiny little blue box right here. The single entire cell would occupy 30 petabytes of data, okay, just for that one single cell at that resolution. Now, if we back off our resolution demands a little bit, say we don't need near atomic resolution of the cell, we only want sort of molecular resolution of the cell, so about 2 nanometer resolution, so where you can begin to see at least tertiary structure, you can see how things are fitting together. Then we get a much larger region, but we're still talking about 30 terabytes just to represent one single cell. So cells are really extremely complex. So when we're looking at cells with tomography, we're really only looking at very localized regions of the cells. So that sort of takes me to this next project. So this, I didn't try to put together proper acknowledgement slides. I'm trying to do the acknowledgements as I go here. This is just from the cover page of the paper that just came out in Cell a couple of months ago. So this was a humongous collaboration between, first started with Mike Rout and Chris Akey, Elizabeth Villa and I joined into the project some years ago. Andre Sally was involved. So this just involved a tremendous number of people, and we're looking at nuclear pore complexes. So this paper used 2 distinct methods. We did single particle analysis and we did subtotal averaging, both on the same NPCs. So if you look at the single particle analysis structure, it's the first time anyone achieved subnanometer resolution using single particle analysis on these large nuclear pore complexes. Now, it's not subnanometer resolution everywhere, as you can tell. This is a gigantic complex. You can see the scale bar here, 200 angstroms, right? This entire complex is spanning, a tenth of a micron or something like that. It's a very large complex. So you can see that the more organized domains in the middle there, you can see them a little bit better in here, were resolved very nicely to high resolution. The sort of scaffolding is very self-consistent and very well resolved. Some of the other domains up here, not nearly as well resolved in the single particle analysis structure. But still, this gives you enough context that you can begin to dock in high resolution crystal structures, or if you like, alpha fold models into the structure, and you can really see with a lot of accuracy what's going on. So you can actually build now a model of the structure of what's going on in this entire complex. Again, very complicated. Don't have time to talk about it in any depth today at all. So the next thing I want to talk about is then the cellular context, and I'll explain exactly why this is critical in a minute. So the NPCs we're looking at here are yeast NPCs. The previous project I showed you, looking at the bacterial the drug efflux pumps, you could plunge freeze the entire cell, right? Because prokaryotic cells, they're much smaller, only a couple of microns in size. So you can just plunge freeze the entire cell. You can do tomography, and then you can pull out the molecules, and everything's beautiful. But if you want to look at larger cells, then you really need to have some thinning technique. So you can make the target thin enough that you can actually image it. So we're looking at here in the upper left, those are yeast cells which have been frozen on a grid. You can see they're sort of clustered together. And then we're putting these into a cryo-FIB mill, focused ion beam mill, originally developed for material science purposes, but now being used for biological specimens as well. So the idea is you take this cluster of cells and then you mill away the stuff that you're not interested in looking at, leaving behind only this very thin lamella. And then you can put the lamella into the TEM and you can do tomography on the lamella. So here's the sort of the overview of all the different cells. You can see the outlines of the cells there. A little bit higher magnification. Now you can see the entire nucleus. Of course, we're only looking at one thin slice of the nucleus. And now here, these are the actual sorts of tomograms that we're using to solve the structure. So you can see here is the double membrane, and it comes up. There's one of the nuclear pores. There's another one of the nuclear pores. And we're just looking at this in the side projection view here. So from using this technique, we can solve an in situ structure of the same NPC. So here's the isolated NPC structure done with single particle analysis up here. And then here's the in situ structure, much lower resolution. This is not achieving the same sort of sub-nanometer resolution that we achieved up here. This is more like 25, 30 angstrom resolution. However, you notice some differences. For example, up here, these NUP82 complexes, which just came out as sort of blurs in the single particle reconstruction, come out much more nicely structured down here. And down here, we can begin to see the basket anchors, which are important in how the channel actually functions. So if you think about this, when you're doing single particle reconstruction of these purified NPCs, they have to be purified very gently. But this is a double membrane spanning protein, right? So you've got the inner and outer nuclear membranes, which are sort of coming in here on the side. And once you've purified them up here, we're really losing that sort of whole membrane context. So if there's any disturbances in the membrane or any structure in the membrane, which is important, we're kind of losing that. So that takes me to the next slide, which explains why context can really be so important here. Now this is from the subtomogram averaging data, from the in situ data, and this is just showing three different little movies of the channel in three different orientations. So this is sort of looking down from the top of the channel. We're just looking at, you can see we're just looking at a quarter of the entire NPC here. And then here we're looking at its side. So these are the two membranes coming in and then sort of crossing over here. And you can see that not only is there a lot of variation in the thickness, we can actually see sort of variations in the associations of the membranes with the complexes here. And you can see how it influences the structures as well. Now this sort of flexibility and dynamics was not part of that paper, but is something that we're now going back and taking a much closer look at. You can see what sort of effect this has on the overall structure of the NPC. So again, putting things in context is very important. All right. So the last part of the work, I'm going to talk about sort of the new technology that we've been developing software-wise to try and address some of this structure, conformational and compositional variability that we see both in single particle analysis and in subtomogram averaging. So this was really the primary work of Muyuan Chen when he was a graduate student in my lab. He actually just moved to SLAC a couple of months ago and is interviewing for faculty positions various places right now. Just want to point out that this concept is actually pretty old. So this is data dating back 15 years now, 16 years now. This is single particle data from before the resolution revolution that occurred about a decade ago. So this is low resolution single particle data of fatty acid synthase. And if we do a single particle reconstruction of that, we can overlay it with a low resolution crystal structure, and it all agrees very nicely. But then when we go in and we look at variability just in 2 dimensions on this, we'll see that this molecule just moves all over the place. It's got over 30 angstroms of motion that's going on in the molecule itself. So this idea of characterizing variability in solution has been around in CryoEM for a very long time. And there have been a lot of methods that people have devised to deal with it. 2D is fairly straightforward. The real difficulty comes when you're trying to do things in 3D. Now, discrete variability is a relatively straightforward problem in most cases. You can deal with it by classification. So say if you have a ligand binding event, you can classify the particles into groups with the ligand and groups without the ligand, and then solve the structures of the 2 pieces. But if things are undergoing this sort of continuous motion, it's a lot trickier. So the approach that we've now developed over the last few years is this Gaussian mixture model approach. So Gaussian mixture models are used quite widely in the modeling community, for example. So Andre Saleh's group, that's one of the big representations you can use there, which is basically just to say that you break down large objects into rotationally symmetric blobs, Gaussians, and you can do this at any level of detail that you like. So you can have a very low resolution representation, or you can increase it. You could have one blob per atom if you'd like. So it allows you, it gives you this sort of very controllable gradiated scale of detail that you can look at in the structure. But the mathematical model is obviously very trivial. You basically have 5 parameters for each Gaussian. You have its XYZ location, you have its width, and you have its amplitude. We can then compare whatever structure we get back with any individual particle. So we can immediately sort of establish that we have this sort of energy function. So if we have a particle and we know its overall orientation, we can make a projection of the Gaussian model in the same orientation, and then we can measure how similar they are. And the more similar, the better, and the better our model is matching the data. So the problem that we have, of course, is that any 2D particle that we're looking at only gives us 2-dimensional information. We're looking at projection images, so it only gives us information in that imaging plane. We don't have any information coming in from Z. So if you try to use traditional linear algebra sorts of approaches to try and solve for the dynamics of the system, it just doesn't work very well. It's also true of tomographic data, because the tomographic data is missing, still missing a lot of Z information. It doesn't have isotropic information as well. So the approach that we've adopted here is to apply deep learning to the problem and allow the nonlinearity of the deep learning process to sort of learn the structural, the conformational and compositional changes that are occurring in the molecule, and give us a latent space that we can analyze further. So the basic idea is the output of this network is one of these Gaussian mixture models. So we have M Gaussians, 5 parameters per Gaussian. So for the output of the network is basically directly comparable to individual particles. The input to the network, so to sort of simplify things, rather than using the entire image as an input, which would make a hideously complicated and computationally challenging network to deal with, we actually take gradients based on sort of a neutral starting model that we start with. So you take the starting model, you compute an energy gradient with each parameter versus each particle, and then that becomes the input to this overall neural network. And then the network training process produces this much smaller latent space in here. Typically, this is a 3 or 4 or 5-dimensional space, and that latent space then might represent all the variability that's present in the system. So I'm just going to show you one example really quickly here. This is from a publicly available data set. This is the ribosome L17 depleted intermediates. And this was the original published analysis that was done. So they basically took their 132,000 particles, and then they classified them into groups, and then they took each of the classes, and then they subclassified each of those classes into subgroups, and they eventually wound up with 18 different submitted structures. But this was a tremendous amount of computational work, because it involves classification and subclassification and sometimes sub-subclassification, and it involves throwing away large chunks of your data, and really a human is sort of closely involved in the process. So we basically took this entire data set, the entire 132,000 particles, and just threw it at the neural network to see what would happen. And the result was actually quite nice. So what we're looking at here in the center, this is the latent space representation. So each of the points in this little map is one particle from the data set. Now, the original classification was 4-dimensional, right? So we produced a 4-dimensional subspace in the neural network, and then we further reduced that down to 2D just for nice visual presentation. But what we're looking at, the classification is actually could be done in a 4-dimensional space. So just to do a very simple initial classification of this, we just split it up into the six sort of most prominent groups. But you can see if you look at it that there's a lot more detail there that we could have extracted as well. And if we look at the various substates that we found just automatically via this analysis, it matches very nicely with the published data. Further, we can also see other things like, I'll let this finish for a second before I switch to the next slide. This is just sort of taking you through the various substates. So the other thing that we found is that there's no guarantees with a nonlinearity of a deep learning technique that directional motion in one part of a latent space is really going to correspond to directional motion in another part of the latent space. However, we found that it actually did pretty nicely. So as you move the ball from one part of the latent space to another, you can see the same helix is sort of growing in and out of the structure. So despite the fact that this is a different state, a different has different ligands bound to the ribosome, that this motion across the state corresponds to the same sort of dynamic change within the map. I'll just toss in one last little example here. This is a precatalytic spliceosome, and this has a more continuous motion. So the precatalytic spliceosome, and when you throw this analysis at that, you can see we get these two different overall motions of the structure out. One of the two, which was identified in the paper when it was published, and then the other one seems to be a novel motion that was identified by the deep learning approach. All right, so that's all I was going to show you for the current state of sort of the deep learning approach. What I showed you with the deep learning based Gaussian mixture model at the moment is still limited to working with single particle data. We're beginning to see that change. So Muyuan actually just came up with a new version of the routine. One of the biggest limiting factors is basically the memory on the GPUs that's being used to compute the deep learning model. That each Gaussian that we had a representation of required sort of an entire image representation in the data, and you'd sort of limited the number of Gaussians that you could use in the representation and the number of particles that you could process at once. When you're dealing with tomography data, you have 20 to 30 times as much image data for each particle, and it just wasn't possible to deal with in the original infrastructure. So we're now working on building on this new strategy that Muyuan put together with some mathematicians at Yale, which is a little bit more memory efficient and can begin to work with the CryoET data, so we can begin to look at the sort of variability as it exists within the cell. The other approach that we want to other thing that we want to do with these Gaussian mixture models is increase the number of degrees of freedom or improve the resolution. So ideally, if you'd like to be able to take this to the point where you could say have each amino acid be a Gaussian, or potentially you could even do each atom be a Gaussian. But of course, if you do that, then the number of degrees of freedom just blows up, to near infinity, because each side chain then, for example, would be rotating independently and need its own degree of freedom to process. So we're beginning to push towards that higher resolution regime and much more complicated analysis of the data. I did all of my acknowledgements during the talk, so I will just stop there and take any questions.
Okay, Steve, thank you very much for these excursions on multiple examples with the technology that you have developed. It's very impressive. So let's see, if any have questions, please put that in your question and answers. And potentially I can actually try out perhaps the mute. Maybe I cannot, okay. All right, one of the questions said that he works on the TOR-B dependent transporter. The protein in the outer membrane is not overexpressed as in TOR-C system. What are the options for him to study the outer and inner membrane component protein together other than tomography?
The protein isn't overexpressed. Sorry, I'm not sure I understood what, I'm not sure I understood the question. Can you say it one more time?
Okay, so if you're looking at the Q&A.
Yeah, I can try and pull it up here. It's not, sorry, it's coming off my screen here, unfortunately.
Okay. Okay. So
yeah, I can't see it.
You can't see it. Okay. So these attendants work on the TOR-B dependent transporter.
Yeah.
The protein in the outer membrane is not overexpressed as in TOR-C systems. So that's a different system.
Yeah.
And he's asking what are the options for him to study the outer and inner membrane components of protein together other than using tomography.
I'm not sure why he wouldn't want to use tomography in that situation. That seems to me like tomography is probably the way to go in that approach.
Right.
I mean,
otherwise, he can do the reductionist approach, purify each of them.
Sure.
Single particle.
But I assume that's what he's already trying. So I'm not quite sure what.
Okay.
Sorry.
All right. Yeah. Okay. We may discuss offline about this particular problem. And there's another attendee asked the questions that he started his journey in crystallography with 3 uncharacterized periplasmic proteins. Among the 3 proteins, he can crystallize 2 of them. One of them only give low resolution defection to 4.5 angstrom, while the other 2 give 3 angstroms. Okay. And he's trying to get a higher resolution data for the others. Meanwhile, to solve the structure of the later, which we collected to 3 angstrom. The space scope of this is P212121. And he's currently going through the molecular replacement using AlphaFold model, which has been promising and productive. According to the cell content analysis, 7 molecules of protein were expected in the asymmetric unit. But the best solution with the decent LLG and TFZ score contain only 2 molecules in the solution. After building 2 molecules of the density, I see a lot of subunit green density, and R work and R3 are high after multiple rounds of refinement. The work continue and appear to be heading toward the right direction. Well, he's okay. I'm reading his question.
I know, I understand. Sounds like a crystallography question. I'm not.
Yeah. Okay, but my screen somehow truncated the question, so I don't know what his question was. Okay, I apologize.
Yeah, okay.
So maybe he can stay on until the end, and he can verbally articulate his questions.
Okay, yeah, okay. The introduction I got, yeah.
Right, okay, let's move on to the next one, which is hopefully will be shorter. What's a typical memory GPU required for GMM with CryoET data?
Okay, well, so with CryoET data, I would say it's not working just yet. I mean, we've got prototype, which is kind of beginning to work now, but it's not quite there yet. There are a lot of issues to deal with tomography. The issue is that when we're doing the single particle analysis, the group size that you're using, in other words, the number of particles that it's considering at one time when it's training the network, it has to be relatively small. I think 20 or 30 is sort of the typical group size that we're using. And if you don't have multiple particles with training the network at once, it just doesn't train very well. So if you think about it in the context of tomography, that would mean that if you wanted to have 30 particles and each one has 30 tilts, means you'd need 900 images at a time, that sort of thing. So the change that Muyuan made recently allows us to increase the number of simultaneous particles by about an order of magnitude. So it's getting into the regime where tomography might start to work, but I think it's probably going to be a few months before we're really ready to say anything. The GPU memory, typical GPUs will tend to vary between sort of 8 and 24 gigabytes of memory right now. You could, if you really get the high end things, you can put together one with like 48 gigabytes. And that's only it's not a big enough factor to really make a big difference. So it's not really all that different if you have an 8 gigabyte model versus a 24 gigabyte model. that factor of 3 doesn't buy you enough to cover the factor of 20 that you need for tomography.
Okay. All right, good. Another question is, how does your analysis compare to the method that is used in the CryoSpark?
So CryoSpark chops the structure up into little pieces in a somewhat automated way, and then tries to look at variations of the pieces. So that, there are sort of 3 kind of general strategies that groups are using to try and build these sorts of energy spaces for looking at flexibility and dynamics. You can just take the structure itself and mask out various pieces of the structure and then try and classify based on that, or try and do local motion based on that. You can just sort of treat the entire system as an energy minimization problem, with the image itself as being the space of the energy analysis. So that's more like some of what Joachim Frank has been doing with the ryanodine receptor, that sort of thing. And then there's an approach like ours, which predefines some mathematical form, which is going to be used to sort of constrain the model, so that having the Gaussians as an intermediate is a third approach. We find the Gaussian approach attractive because it allows you to define the level of detail at which you want to do the analysis, and it allows you to sort of block out specific regions and say, okay, I want to allow these Gaussians to move, but I don't want to allow any of the other Gaussians to move, without having to build these sort of masks around different parts of your structure. So anyway, that's sort of the general difference between the approaches.
Okay, thanks. Can you briefly explain how you train your model, and also explain how you give a physical meaning and interpretation to the latent space?
Okay, so the training of the model, I skipped those slides just in the interest of time. But the model is trained in 2 stages. You've got the encoder and the decoder. It looks very much like an autoencoder, although it's not quite an autoencoder. So you start by training the decoder with a neutral structure, which sort of defines the center of the latent space. So that's supposed to be just sort of an average structure, including all of the data. And once you've trained that, then you incorporate the encoder as well, and then you train the entire system. But you're training it against particle data. So you're basically putting the entire set of particle data, and you just iteratively train it against the particle data, trying to reduce the difference between the model and the individual particles. Sorry, what was the second part of the question?
Yeah. So
meaning on the latent space. I remember. Yeah. So assigning meaning in the latent space. So that's actually one of the really nice things about the Gaussian mixture model, is that when you saw that map in the latent space, you could sort of immediately just sort of mouse over any portion of the latent space, and you can immediately see the Gaussian representation. If you look at the other sort of latent space kinds of schemes that people have been developing, generally what you have to do to assign meaning is you have to go into a portion of the latent space and draw like a little circle, basically, and then you pull out all the particles in the circle, and then you have to do a 3D reconstruction of all the particles. So the maps I was showing were actually reconstructions from particles like that. But with the Gaussian mixture model, you can actually immediately see the Gaussians moving. I mean, you just put it through the network and you get the Gaussian output, which is a visually interpretable model, before you spend the hours or days that it's required to regenerate all of those 3D reconstructions.
Okay, so the next question you might have already answered it, is how your method different or similar to the CryoDragon software?
That's sort of what I was answering. I mean, the CryoDragon software doesn't use a Gaussian mixture model. It uses a different mechanism for representing the re-representing the data. So it doesn't go through one of these intermediate states. So in CryoDragon, if you want to get results out, you have to do these local reconstructions if you want to interpret the latent space. But otherwise, they're similar in a lot of ways.
Okay. So another questions related to the data size. what is the minimum number of particles required per stage for the Gaussian model method? If you want to go into atomic resolutions, how many particles you need?
All right, so that's basically the same as asking how many particles do you need to do an atomic resolution reconstruction. The idea is once you have your latent space, if you pull out particles from a large region of the latent space, then you get more particles, and if you use a small region of the latent space, you get fewer particles. Obviously, if you have in the smaller region of the latent space, you have a more self-similar subclass, right? So in other words, you can add, as you add more particles, you're adding more variability at the same time. Typically I would say you'd like to have, say, 100,000 particles when you go into the analysis if you can. You can do the analysis with 10,000 or 20,000 particles. If you're just looking for discrete variability, a ligand binding event or something like that, then you just need sort of twice as much data as you'd need for a single atomic structure. And then you're classifying it into two groups. But if you're looking at continuous variability, then it depends on the level of detail you want to look at the variability. I mean, with say alpha helical resolution, I'd say you probably need maybe 30 or 40,000 particles. If you want to do near atomic resolution and really chop it up into a bunch of groups, then a few 100,000 is probably a good idea.
Okay. And when you talk about the Gaussian feeds to the blobs, you mentioned energy functions. Can you explain what do you mean here? What kind of energy you refer to?
Well, so you have to have something to minimize as you're or maximize, depending on how you look at it, as you're training the network. So what we're using as a similarity metric is just the Fourier ring correlation between the model and the individual particles, and then you sum that over all of the particles. So that's what we're using as an energy function. And there are a bunch of advantages to using that. Yeah, that's what we mean. It's not a true energy in any real sense. It's a energy from a computational minimization perspective.
Okay, so there are a couple of questions. Was the CryoEM used to help isolate the nuclear pore complex of interest, or were in-situ complex chosen and not overexpressed?
Yeah, there was no overexpression. These are just native yeast cells. So that means we got very low yield. Given the size of the complex and the resolution that we were targeting, we typically get 1 to 3 or 0 to 3 nuclear pores with each tomogram. So that published paper, there were 1,000 tomograms. So several hundred lamella, 1,000 collected tomograms, and about 600 particles.
Yeah, right. So the questions I think raised in here is whether you could apply the deep learning algorithm to focus on particle of interest, and that particle has been tagged with some fluorescence tag. I guess, in other words, how have you tried to apply your method with particle of interest that has been tagged by fluorescence? In other words, in your nuclear pore example, you just look at the nuclear pore, you get what you get, right?
Sure.
You are not particularly hunting for the nuclear pore at the beginning, maybe.
We're not hunting for specific nuclear pores in specific states or something like that. I mean, if you fluorescently tag a nuclear pore, the resolution of the fluorescence is pretty, ClEM is useful for sort of localizing regions that have the protein of interest, but it's not at the level of an individual nuclear pore. The nuclear pore is smaller than the wavelength of light. So I'm not sure what relationship with fluorescence tags they're talking about.
Okay, maybe we can get it together an hour from now. Okay, Steve, thanks very much for your excellent presentation. And For those who have questions, please stay on after the second speaker. So now I turn it back to Bob.
Thanks, Wah. Thanks, Stephen. Great, very interesting talk. A huge amount of data for one structure analysis. So now we'll go on to the physical sciences talk. Professor Yi Che will introduce Professor Jaeger. Apparently we may not be able to record all the talk, so we'll go through the introductions and take it from there. So please, Yi.
Yeah, thank you, Bob. It's a great pleasure to introduce Professor Wolfgang Jaeger from University of Kiel. Well, Wolfgang got his education and then early appointment in US and Germany. In 1996, he was appointed as a professor and director at the Institute of Material Science of Christian Albrecht University in Kiel. Well, he certainly contributed a lot to building up the Kiel, the university's infrastructure in the course system. So today, Wolfgang is going to tell us about his advanced in-situ TM study for the development of high-efficiency solar cells. Wolfgang, take it from here.